Hello, everyone. Here we are in the last part of this chapter. We have covered replication and transcription. Now we're going to cover translation and hopefully regulation. Okay. So where are we at? Translation. We have made the messenger RNA. Now we have to turn that messenger RNA into a sequence of amino acids to make a protein. That's translation. So the first thing that we have to start out with here is the concept of the genetic code. Very, very neat thing. Very cool thing. Okay. This was elucidated by effectively two researchers, and they came up with the conclusion that the genetic code consisted of what's called a codon. In other words, a small word of nucleotide. And we found out that the words were 3 letters long. Okay, so every codon is three nucleotides long. And so the next question is, well, how many combinations of 3-letter long words can you make if each position has a possibility of 4 nucleotides, guanine, cytosine, thymine, and adenine? And the short answer is 4 times 4 times 4. Which is 64. So here's a genetic code with the 64 codon combinations. Okay. So you can see that UUU, uracil is phenylalanine. UUC, also phenylalanine. UUA, uracil A is leucine. So what this tells us is that the genetic code is degenerate. What this means is that more than one codon codes for the same amino acid. How many amino acids? Hopefully you remember that there's 20 amino acids. So if you look here, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. I missed one somewhere, but 20 amino acids, okay? There's also 3 codons that code for a stop. 3 of them, UAA, UAG, and UGA are stop codons. That tells the translation event to stop. And there's also a start codon. There's only one. That's AUG. And AUG codes for the amino acid methionine. So this is interesting because this tells us every single protein that you have, every single protein that I have, every single protein that animals have, plants, fungi, bacteria, you name it, they always start with the amino acid methionine. Very interesting. Okay. So here's our genetic code, 64 degenerate codons that code for 20 amino acids, 3 stop codons, and 1 start codon. Okay. Now, as I said, the name of the game here is to go from messenger RNA to protein. So what I'm going to do, I'm not going to go into any of this stuff. It bores me. What you need to know is that here's a messenger RNA. Okay, you need to know the machine responsible for this event. Remember, the machine for DNA replication was DNA polymerase. The machine for transcription from DNA to RNA, that was RNA polymerase. And the machine for translation from messenger RNA to protein is this beautiful guy called a ribosome, okay? So a ribosome consists of 2 units, a small and a large subunit. The large subunit has 3 sites that are important to know. In order, it is the A, the P, and the E site, okay? The A or acyl site, because the acyl site of the amino acid goes in there. The P for peptide, because that's where the amino acid peptide bond is formed. And E for exit. So again, the sites are A, P, and E in that large ribosomal subunit. Now, surprisingly, there's 3 stages to translation: initiation, elongation, and termination. Okay. So in initiation, you have the messenger RNA that just got produced. You have the small ribosomal subunit. You have the start codon. Remember, in the genetic code, we have one start codon, AUG. Here's the anticodon on the so-called transfer RNA. And here's the anticodon, and here's the amino acid. Now, in prokaryotes, it's not just methionine, it's 4-mil methionine, but effectively it's a different type of methionine. So these two find each other because they're complementary, the codon and the anticodon. Energy is expanded in the form of GTP to GDP that causes a change in the conformation of the small subunit that causes a large subunit to associate. And notice that only the P site is occupied. There's no A or E tRNAs in place. This event that consists of the messenger RNA, the small ribosomal subunit, the large ribosomal subunit, and the start transfer RNA with the anticodon to AUG and the 4-mil methionine, that is called the initiation complex. Okay, so that's the initiation. Now we're going to go to elongation. Okay, so this thing chugs like a little train. All right, and what you need to keep in mind is that there we go. Is that we started with a start codon on the P side, and the A side is going to be for the assimilation of additional tRNAs, and the E side, the X side, is going to be for the removal of tRNAs. Okay, so notice that here we have the tRNA that's complementary to the codon UUU with phenylalanine, just like we said at the genetic code. Okay, here in the P side, we see that we are forming a peptide bond between the first and the second amino acid. When that happens, notice that the covalent bond between the amino acid and its tRNA is broken. See how this is here, but it's not here? So this amino acid has now dissociated from its tRNA, and this tRNA is consequently empty now. Okay? Now, the ribosome is going to move 3 nucleotides or 1 codon. That's called translocation. Okay? So the phenylalanine is going to move from the A to the P site, and the empty F-met tRNA is going to move from the P to the E site, leaving an empty A site. This guy, because it's empty, is going to dissociate out of the X site. No surprise there. And now this A site is there, accessible for the next complementary codon to come in. Very, very neat. So that's what keeps on going on and on, okay? This guy comes in, this one goes out, this one moves over 1 codon or 3 nucleotides, and the process continues, and you continue to make a polypeptide chain. There it is. Okay, what's left? Well, termination. We have termination. So in termination, it's not showing it, but effectively in termination, what we're going to have is that we're going to have a stop codon here. That stop codon is going to get a modified tRNA that does not have an amino acid. When that modified tRNA comes in and hydrolyzes, it causes this amino acid chain to dissociate from the last tRNA, and then the entire complex falls apart. So that's termination. Okay, very neat stuff. Let's see. And let's move on to regulation. Okay, so again, regulation is either causing a gene to be expressed or causing a gene to not be expressed. In other words, we're regulating if a gene gets expressed. Somebody is pressing on the gas on how often a protein gets produced. So in prokaryotic organisms, the classical models that we look at regarding this regulation are called operons. And classically, we look at the LAC and the TRP operon. LAC stands for lactose, and TRP stands for tryptophan. Okay, so let's look at the big model here. What does an operon look like? Well, you're going to have a promoter region in a regulatory gene region that causes causes the production of a repressor. Okay, the production of a repressor. Downstream, we have another promoter, and in front of it, we have something very important called the operator. That's why this whole thing is called the operon. This is the key protagonist in this entire event, the operator, okay? And then downstream from there are the genes that are either going to be expressed or be subdued, okay? So let's look at, okay, one point to make is that in prokaryotes, you either have inducible or repressible operons. So inducible means to induce. That means that the operon is shut off, but we can induce it. We can turn it on. Or you can have a repressible, which means that the operon is on, but we can repress it or turn it off. Okay, that's it. There's no other choices. So as it turns out, lactose is an inducible operon. Okay, why is it inducible? Because microorganisms are not typically around the sugar lactose. The first sugar that any organism, including ourselves, breaks down is glucose. We'll break other sugars down once the glucose is gone. Once there's no more glucose, we have no choice. We have to go to other sugars, okay? But it would be silly for us to express the genes necessary to break down lactose if there's glucose around, because it takes energy to make the message and it takes energy to make the proteins. So why invest all this energy in catabolizing a sugar that we don't need to catabolize because glucose is around? Now, once glucose is gone, things are different. Now we have to switch to a different sugar. And that's where the lac operon comes in. So what this means is that the lac operon is typically shut down because we're breaking down glucose. When we run out of glucose and lactose comes in, now the bacteria and even ourselves go, uh-oh, it's time to make the proteins necessary to catabolize lactose, okay? So here's our operon, okay? Here's the promoter for the regulatory gene. And this regulatory gene carries out transcription and then translation to make a repressor, okay? Here's my repressor protein. Now, under normal conditions, we have sugar, we have glucose and no lactose. So what that repressor does, it associates with the operator right here. And when that happens, it prevents RNA polymerase from associating with the promoter and transcribing the genes necessary for lactose catabolism. So that's the main point right there, okay? We have a regulatory gene that produces a repressor that is functioning. How do we know that it's functioning? Because it's doing its job of getting in the way by associating with the operator. And when that repressor, imagine a train track, okay? And in order for a train to go down the track, it has to get on the promoter and go down the track. But it can't do it because there's this garbage in the way called a repressor attached to the operator, and it blocks the engine from going on the tracks. It blocks the RNA polymerase from going on the tracks. So because of this, RNA polymerase cannot attach to the promoter and nothing happens, okay? So when does it get induced? Well, when we have run out of glucose and sugar comes in, what kind of sugar? Lactose. So lactose is called the inducer because that's the presence of the lactose sugar is what induces or activates this gene. So the inducer, allolactose, that's produced from the sugar lactose, it associates with the repressor, and what it does is it turns it off. It turns the repressor off. If the repressor is off and not doing its job, that means that it no longer attaches to the operator. If it doesn't attach to the operator, it means that RNA polymerase can now attach to the promoter and transcribe the genes necessary for lactose catabolism. That's it. That's a lac operon. Okay. There's more to do here with CAMP, but I don't want to bother you about that. Let's move over to trip operon. Here's a trip operon. Okay. So trip is tryptophan. Tryptophan is one of the 20 amino acids. And the problem with tryptophan is that it is a very large, very complex amino acid. And it is so hard to keep up with demand of this very large, very complex amino acid. So what happens is that we need to have the gene on all the time, producing tryptophan so we can incorporate it into our proteins. But every once in a while, we catch up and we end up with too much tryptophan. Uh-oh, now it's time to slow down. So the trip operon is a repressible operon because typically it's in the on position, and the presence of too much tryptophan represses it or slows it down or shuts it down. Okay? So what is different? Notice we have, our author didn't put this in yellow, shame on them, but this is supposed to be the promoter for the regulatory gene. And here's a regulatory gene. And then that produces a messenger, transcribes a messenger RNA that produces a repressor. And here's where things are different. Notice that in the lac operon, the repressor is active. And because it's active, it attaches to the operator. However, in the trip operon, the repressor is synthesized in the inactive form. In other words, we make it, but it doesn't do anything. And because it doesn't do anything, it doesn't do its job, where its job is to attach to the operator. So if it doesn't attach to the operator, RNA polymerase can attach to its promoter and synthesize the enzymes necessary for the manufacture of tryptophan all day long. But like we said, every once in a while, we catch up with demand. We end up with too much tryptophan. And when that happens, the tryptophan, which is called the co-repressor, just like allolactose is the inducer because it induces the gene, the product, tryptophan, is called their co-repressor. And when it associates with the inactive repressor, it turns it on. And when it turns it on, it does the job it's supposed to do, which is to associate with the operator. And when it associates with the operator, the RNA polymerase can get through, and it effectively shuts down the trip operon. That's it. Not too bad. Now you've learned about prokaryotic regulation. Hope you're having a wonderful day, and I'll see you guys soon.